Okay, 15.2, gametogenesis in humans. The necessity of gametogenesis. The production process of reproductive cells or gametes is called gametogenesis. So this process to produce gametes known as gametogenesis. This process takes place in the gonads, which are the testes in males and the ovaries in females. So the location will be in the reproductive organ. Okay, the process take place in the reproductive organs that are testes and ovaries. Gametogenesis produce gametes that are haploid. Okay, so remember what is the meaning of haploid? Okay, so gametes are haploid. When fertilization takes place, the nucleus of the sperm will fuse with the nucleus of the ovum in the fallopian tube to form a diploid zygote. So when it fuse together, sperm and ovum fuse together, it will produce a diploid zygote or 2N. So zygote is 2N lah. Okay. So we proceed with the first one, spermatogenesis. Okay, spermatogenesis is a process of sperm production that take place in the seminiferous tubules. Figure 15.3 shows a longitudinal section of the testes. Okay, so we have males have testes, right? So the testes usually like this lah. I draw just like this, right? So we have the testes, two testes here. So if you cut this longitudinally, you will get this diagram lah. Okay. So let us see the structure first. So the structure, we have the seminiferous tubules. So we have this one, seminiferous tubules. Okay, a lot of seminiferous tubules here. And then we have the epididymis. Okay, and then sperm duct or vas deferens. Same meaning lah. Sperm duct of vas deferens. So what is inside here? So we have also this one, blood vessel lah here. Okay, right? So each seminiferous tubule consists of primordial germ cells. So inside here, seminiferous tubule have germ cell. Okay, so I just draw a germ cell here. Okay, in the seminiferous tubules, okay, germ cells. So primordial germ cell will undergo cell division to produce sperm. So this cell function to produce sperm. It will undergo cell division. This means it will undergo mitosis and meiosis to produce sperm. Okay, so it starts with this cell, primordial germ cell inside this seminiferous tubule. So Sertoli cells within the walls of the seminiferous tubules provide nutrients throughout the process. So another cell here at the wall here, we have another cell known as Sertoli cells. Okay, Sertoli cell function to provide nutrients throughout this process. So there are two cells inside these seminiferous tubules, primordial germ cells and also Sertoli cell. So primordial germ cell will undergo cell division to produce sperm, whereas Sertoli cell just produce nutrients lah. Okay, provide nutrients for this process. So then after process occur, produce the sperm, right? So the sperm then will be flow into this epididymis. Okay, epididymis here. Okay, and then we go to this epididymis. This one is the head of epididymis lah. Then to epididymis, and then will be transported to the into the sperm duct here. So this is all the sperm lah. The swim here lah. Okay, to the sperm duct, and then will be flow out lah to the urethra. Urethra inside the penis. Okay, you still if you not remember, you just go back and look at look back the diagram, the female reproductive system. Okay, so this is the structure lah of the seminiferous tubules. Okay, spermatogenesis is stimulated by the follicle stimulating hormone FSH and testosterone. This process is also aided by the luteinizing hormone, which stimulates testosterone secretion in the testes. So this process need hormones. So we have learned before this in chapter 12. Hormones usually used in the reproduction. So in this case, we have the FSH and also testosterone and LH. So, but the LH function to stimulates the testis to produce testosterone. That means testis cannot secrete the testosterone on by itself. Okay, it must be stimulated first by the LH. So then it will produce this testosterone too, and also at the same time with the FSH to stimulate the spermatogenesis. Okay, so you must remember the hormone that involved in this spermatogenesis. Okay, FSH, testosterone, and also LH. Okay, so let us take a look at the process. What about the process? Okay, so process here we have meiosis and meiosis. So start with the multiplication phase here. Start with the primordial cell. So this one is primordial germ cell. So one cell. We divide mitotically to form diploid spermatogonium. Okay, so this primordial germ cell will divide repetitive mitotic division, okay, to form the spermatogonium. So still 2n because mitosis or mitotic. Okay, the number of the chromosome number, right, same with the parents. So that's why this one also 2n. So we name is for example as spermatogonium. Okay, so actually divide. So a lot of the spermatogonium will be produced lah. Okay, then spermatogonium expand to form primary spermatocyte. Then it will enter the second phase, that is growth phase, whereas this spermatogonium will develop into primary spermatocytes. One spermatogonium here will develop into spermatocyte. That's why this one 2n, this one also 2n. Okay, no changes, no dividing, just develop only. So the number, chromosome number still same lah. Spermatogonium also 2n. Primary spermatocyte also 2n because just develop only. Okay, diploid, expand. Just expand to form spermatogonium, expands to form primary spermatocytes. So diploid. Okay, then what happens next is that go into the next phase, that is maturation phase, where in this case each primary spermatocyte undergoes meiosis 1 to form 2 secondary spermatocyte haploid. So this primary spermatocyte will undergo meiosis 1 to produce 2 secondary spermatocyte. So now secondary spermatocyte is haploid. Okay, because meiosis 1, still remember meiosis 1 half the number of chromosome. Okay, and then each secondary spermatocyte undergoes meiosis 2 to produce 2 haploid spermatids. So now this secondary spermatocyte, each of them will undergo meiosis 2 to produce spermatid. So we get 4 spermatids, okay, and also haploids. Okay, so you still remember meiosis 2 actually just like mitosis only, same with mitosis. Meiosis 2. That's why the number of chromosomes still maintain lah, n lah. We don't have half n, no. So n. Okay, and 4 spermatids. Okay, and then the next one is spermatids then undergo differentiation to form sperms. Okay, so these spermatids will undergo differentiation to form sperm. So what is differentiation? In this differentiation, some structure will be added to the cell. So in this case, we have tail lah. So now these spermatid have tails, can swim. Okay, and then the structure also a bit like this, right? Not circle. Okay, so this is known as differentiation. So now this structure known as sperm and still haploid. So this one you must remember. Sometimes give the diagram and ask you to label the structure, label what's here. For example, in this, what is the name of this one? You must remember primary spermatocyte. Okay, and then the number of chromosome, diploid or haploid. So you can remember we start with the pre. So if want to make this one, we have the primordial germ cell. Okay, undergo mitosis, produce spermatogonium. So this one is 2N. Okay, spermatogonium just develop, produce primary spermatocyte. So of course still 2N. Then undergo meiosis 1. Produce secondary spermatocyte. So now this one N. Half. Okay. And then undergo meiosis 2. Right. Meiosis 2. Produce spermatids. Also N. Then spermatids undergo differentiation. So produce sperm also and so this one you must remember. So these are the process of the spermatogenesis.